It seems like you got everyone.
Thanks.
Charlie's not here. You emailed me. Studebaker is not here, but he's coming. Is this Minnie?
Yeah, it is.
I thought it was Minnie.
Black and small. Big one.
Oh yeah. This one?
Yes.
Oh yeah.
Your notes are also in there. Let's see. We're looking for them.
Dakota.
Yes, I do eat a single one. I mean,
Really?
Well, I mean, like, eat like 2 cups, but like, I don't eat
Yeah.
breakfast.
You don't have to share with people if you don't want
No,
to.
it's fine. It's just because I wake up so late that I don't time.
Okay.
Yeah.
Yeah, as I was going through it, I'm like, this dude only eats like 1 meal a day. Like, how do you do that? Especially on like a teenager to early 20s metabolism. How do you cut that?
Oh, I eat a lot. Whatever it says in here, I mean, I eat a lot of. So.
You don't eat more than Brandon.
Huh?
I eat a lot too, man.
Can I share your Taco Bell run?
Yeah, I go for it.
Holy crap. What was in your Taco Bell run? Like 8
There's like 10 tacos.
tacos and...
Bean burrito.
No,
Taco box
don't stop there.
Yeah.
There was like then Locos Nachos, and I was like, man, he's tall and everything, but he can pack it away.
I don't know where it goes. Yeah,
Yeah.
it's
Try
like,
different looking.
No, you're not overweight or anything. I was just.
Crunch Wrap Box, the new triple double Crunch Wrap Box. Have you gotten that yet?
I haven't got that. Is it pretty
It's
good?
pretty good, yeah.
I was just shocked to see the volume of a single Taco Bell. All right. I put a link up on the Moodle site for the videos. The video worked last week, so it won't fit our Moodle. The files are too big, so they're just on my YouTube page. There's now a playlist for CTI 259 videos. You can go there and find
I
them.
usually do too.
Is that link just right on Google? Okay.
It's a link to the playlist, and then you'll find the playlist.
Oh,
So,
all?
yeah.
Do you have the charging cord at all?
For
Like iPad?
in my office, I do.
Dang.
Do I need to go get one?
Well, I mean,
for you?
it's dead, it's basically.
It's at 9%.
It's at 9%, and my charging cord won't work, and I don't know why, dude. This is-
Well, do you want to run down to my office and get one?
So I'd love to.
Yeah.
I mean, I do want it to go, so.
You don't want to pencil on a
It's
paper?
gotta be two
It's
posts.
142 next to the,
So we've got.
next to this Yellowstone
Yes.
in Whiteside.
Yeah.
Just go in there with my office, there's a big long table, and there's a power strip next to my monitor. And there are a couple of.
Thank you.
Sure. All right. Refocus. Where were we when we finished up last week? I know we
Cells.
talked about karyotypes. Did we talk about duplicating chromosomes? I don't think we had yet.
Yeah, we did. No.
You haven't seen this.
We
got,
saw that.
I think
Saw
we were
that.
here.
We saw that, yeah.
So we were talking about chromosomes. So before we start, do you have any questions over what we were doing last time? Okay, we're a little behind, but we might or might not get caught up. It's not a big deal if we don't. I'm not gonna rush through things to try and get back on schedule. We'll just take it as it comes. So we were talking about how genes get packaged up. So DNA molecule gets wrapped around histone molecules. Those histone molecules form these globs that then form kind of long segments, and those segments get folded up on one another until eventually they stop looking like long strings of DNA, get wrapped around the histones. Those histones coalesce. Those histones form long fibers. Those fibers fold up on one another until you finally get to a chromosome. And the chromosome is kind of the particulate nature of inheritance, so that you physically inherit these chromosomes that have tons and tons of genes on each chromosome, but they get inherited in this form. They don't get inherited as individual genes that aren't potentially linked to other kinds of genes because they're on the same chromosome. So we can look at these genes as, or these chromosomes as single chromosomes. But remember remember I told you that we could also envision these chromosomes after they've gone through the process of duplicating their DNA, and I was going through on the board showing you that when you start off as a zygote, you're one cell, and then you go to the 2-cell stage, and then you go to the 4-cell stage. Well, at each of these duplications, you have to have duplicated your DNA, otherwise you don't have any DNA to put into the new cell. So if you start off with,
Thank you.
Sure. If you start off with 46 chromosomes here, you have to duplicate this to 96, sorry, 46, 92 chromosomes in order to get 46 chromosomes here and 46 chromosomes here. And so you have to go through this duplicating phase. And when you've gone through that duplicating phase, before you then go through cell division, each chromosome looks like it has 2 pieces. And so it looks kind of like an X. One old chromosome is here and the new chromosome is here. So in order to kind of understand what's going on with this, the terminology is weird because we refer to chromosomes, and this is even confusing for biology majors, so don't sweat it if it's confusing to you. Chromosomes can either be the chromosomes that have one copy of DNA, or they can be chromosomes that have 2 copies of DNA. But in this case, we refer to it as one chromosome with 2 chromatids. So what this is showing is you start off with one chromosome. Let's say that this is chromosome 1 of the 46 that you have, of the 23 different types that you have. Chromosome 1 duplicates itself in preparation for cell division. At this point, we still refer to this as a chromosome, but it has 2 sister chromatids. So this is a chromatid and this is a chromatid. This chromatid was built from copying this chromatid. And so remember that you have 2 homologous chromosomes. You have one chromosome 1 that you got from your mom. You have another chromosome 1 that you got from your dad. This is a homologous pair of chromosome 1, one from mom, one from dad. And this is that banding pattern. You know how when you stain them with Giemsa stain, they take on this banding pattern because of the density of chromatin. And so they're the same chromosomes. This chromatid is genetically identical to this chromatid. This one chromatid is genetically identical to this chromatid, but this chromosome can be genetically different from this chromosome, and that it can have different versions of the genes. So if there's a gene that occurs here for tongue rolling and it's a dominant allele, remember, what are alleles from last week?
That's what you ability to do it or not to do it.
Okay, so it's just different forms of the same gene. So we have a gene tongue rolling, and you either have the gene that allows you to do it, or you have the gene, the version of the gene, the allele that doesn't allow you to do it. So remember that alleles, genes come in different forms, which are alleles. And so you might have the allele for tongue rolling on these 2 chromatids, but you might have the allele for not tongue rolling on these 2 chromatids. They're the same on each of the 2 chromatids because this chromatid was built from copying this chromatid. So the sister chromatids on a homologous chromosome should be genetically identical, but the alleles on one homolog might be different from the alleles on the other homologous chromosome. So we have to get these copies made because then we're going to take one of these chromatids, put it into one cell, take another one of these chromatids, put it into the other cell, take this one, put it into this cell, take this one, put it into another cell, as we make our second cell as we're going through development. You guys look stunned or bored or something. Am I moving too fast? Or is this just old hat for you? Or is it genetics and you just can't stand genetics? Okay, I'm gonna press on that. So, one of the cool things about DNA is that its structure makes it easy to make copies of it. Remember that these nitrogenous bases in the center of DNA, remember this is the sugar phosphate backbone. These are nitrogenous bases. How do these nitrogenous bases bind to one another in DNA? What's the kind of bonds that they make?
Hydrogen?
Hmm?
No, wrong.
No, go ahead.
Hydrogen?
Hydrogen bonds,
Yes,
yep,
okay.
yep, that was exactly correct. Don't question yourself, just go for it. What's special about hydrogen bonds? Somebody else?
Covalent bond.
They're not covalent bonds, they're hydrogen bonds. So how are they different from covalent bonds?
The way that they get, the oxygen.
No, it doesn't have to do with, well, it has to do with a property of oxygen. Remember, oxygen is an electronegative atom. Electronegative atoms do what to electrons of other atoms?
They pull them
They
together?
pull them away. That sets up this polar situation where oxygen is partially negative and hydrogen is partially positive. And then what is that hydrogen able to do? It's partially positive. It can form
And add.
hydrogen bonds with what other kind of atoms? If it's partially negative, it can form bonds with partially positive atoms. So if you have an oxygen, hydrogen is partially positive, it can form with bonds with partial negative atoms. So that partial negative and partial positive form the hydrogen bond. Are those hydrogen bonds as strong as covalent bonds or ionic bonds? They're not. They're relatively weak. And so because these nitrogenous bases are being held together with hydrogen bonds, it's easy to take the two halves of DNA and just split it apart, unzip it, essentially. When we do this, when we make DNA, not with heat, but with an enzyme called helicase, it just is an enzyme that unzips DNA. And what happens is this helicase comes through here and it breaks these bonds, and when it exposes those nitrogenous bases, remember that those nitrogenous bases want to form complementary base pairing with the nitrogenous base that they complementary base pair with. And so what you'll have is when you expose a thymine, you will have an adenine come in here and bond to it. And what it's not showing you on here is that's just not just an adenine nitrogenous base, that's a whole nucleotide. It's a phosphate bonded to a sugar, bonded to an adenine. And when it's duplicating itself, the thymine that comes in next to it will then form a bond here, while it's also forming a bond from adenine to thymine. Forms two bonds if I recall correctly. Adenine to thymine, this would bond with thymine on this strand of the DNA. And then a new bond will form between this nucleotide and that nucleotide. Remember, a nucleotide has a phosphate molecule, the sugar molecule, and the nitrogenous base. So they're not really showing you the sugar phosphate backbone here, but they're showing you the complementary base pairing. So because of complementary base pairing, when you open up DNA is naturally going to want to add new nucleotides to those nitrogenous bases that now have their bonding sites exposed. So adenine will naturally want to bond with thymine, thymine will want to bond with adenine, cytosine to guanine, guanine to cytosine. And they still use enzymes to facilitate all of this and make it more efficient, but there's this natural affinity of these nucleotides, the nitrogenous bases of the nucleotides, to come together. And so if you unzip it, you use the old strand to then help you synthesize a new strand, and the sequence of the new strand is determined by the sequence of the old strand. Because of complementary base pairing, adenine always bonds to thymine, cytosine always bonds to guanine. Okay? So it's really a pretty natural sort of thing to have this structure of DNA that naturally wants to duplicate itself in a sense. And so during DNA replication, when we're making copies of DNA, the molecule is split, new nucleotides are added to the exposed nitrogenous bases. then those are stitched together to complete this sugar phosphate backbone. And each new strand of DNA is made up of half of the strand is original DNA, half of the strand is newly synthesized DNA. And so they call this semi-conservative replication because it's conservative in that you're always using the old template of DNA to create a new sequence of DNA that makes up the other strand. You don't ever create a new DNA strand that is totally new. There's always half of the old hanging out, being used as a template. So think of the DNA strand as a template that you use to build new DNA. I do woodworking periodically when I have time, mostly during the summers. Last summer, I renovated my kitchen and we wanted to put in a butcher block countertop that we were then going to epoxy. But in the countertop, we had this really long countertop. And it was so long that I had to actually put two countertops together to do this. But then we were going to put a farmhouse sink in the middle of this. And so the butcher block tabletop is 2¼ inches, 2½ inches thick or something like that. So it's really, really thick. So I was gonna have to route out the hole in this really long countertop in order to do this. And so I had spent, I don't know, quite a lot of money on the countertop itself so that I didn't want to screw the countertop up by cutting a hole in it to fit the sink in there. So what I did was I got cheap wood, what they call MDF, medium density fiber board, and I put the sink on top of that and I made an outline, and then I cut that out with my router, and then I set my sink down into that cutout to make sure that it would fit. Once I knew that it was cut right, I then clamped it to my countertop, and it served as a template for the router to do the cutting. So I could just push up against the MDF template and then use that as my guide to making the hole with the router. And it worked perfectly because I had a template that was kind of discardable, made out of something cheap, but firm, so that it would allow me to press up against it as I routed. And that template then made me able to create this thing, this opening correctly. In the same way, the DNA template on the original strand serves as the template so that you get the order of nitrogenous bases in the new half of DNA correct. And that's what templates are all about. So when I use this term template, that's what I'm meaning. As we go from, so this has to do with cell division. We need new DNA in order for our cells to divide because we have to duplicate our DNA and our chromosomes and everything during cell division. But we also have to copy DNA in a different way because when we go to build the building blocks of these new cells that are being built, all of the proteins that go into making those cells function, we actually have to take DNA, and from this we have to make a messenger RNA molecule. And from that messenger RNA molecule, we're going to get the order of amino acids in a protein. So this is DNA. It has nucleotides. Remember that a nucleotide is a sugar and a phosphate bonded to a nitrogenous base. RNA is also made up of nucleotides. It's a phosphate bonded to a sugar bonded to a nitrogenous base. But amino acids are the building blocks of proteins. And they're not nucleic acids. They're a different category of biological macromolecule. But we use the order of nitrogenous bases in RNA to specify the order of amino acids in a protein. And we use the order of nitrogenous bases in DNA to specify the order of nitrogenous bases in RNA. So the gene in DNA specifies the order of nitrogenous bases in RNA, and then that specifies the order of amino acids in a protein. So the way we get this done is just by doing another type of copying of RNA. We basically take DNA, and from that we use DNA as a template to make messenger RNA. This process is called transcription. This process of going from the information in RNA to amino acids, this is called translation. And we use these terms because it's kind of like language. If you think back, have any of you guys ever seen the movie The Name of the Rose? Oh my gosh, The Name of the Rose, you guys need to get out more often. Name of the Rose is this movie. It's from a novel by Umberto Eco, if you know who that is. Anybody familiar with Umberto Eco? No? Okay. Sean Connery plays a Franciscan monk who is basically a Franciscan monk version of Sherlock Holmes, and he gets called to this abbey up in the mountains of Italy to basically investigate a series of mysterious murders that is happening at a monastery that has a very famous old library. And so a lot of this plotline revolves around books. Sean Connery and his trusty sidekick, a very young Christian Slater, who is a novice in the order, go there and they try and solve these crimes. So a lot of it revolves around books and how books were made back before we had a printing press. And what would happen is when you needed a copy of a book, you would get a book that you wanted to copy, you would get an empty book that was just covers and a bunch of blank pages, and a scribe would sit in a scriptorium and day after day after day just sit and transcribe this book into this other book, verbatim, same illustrations and everything. And so this process of transcription is taking a document or something in one language and then making a copy of it in that same language. So we talk about transcription because we're taking the language of nucleotides and nitrogenous base sequences and we're making a copy of that that is still in the same language of nitrogenous base sequences. But when we take that and use that to specify proteins, we're translating it because we're leaving the language of nitrogenous base sequences and we're now translating that into the language of amino acids in a protein. So the difference between transcription and translation refers to whether or not we're staying with the language of nucleotides or if we're moving from the language of nucleotides to the language of proteins. So the way this works is the DNA molecule is split just like it's split during the replication of DNA for cell division, but in this case, there's a different set of enzymes that are going to be adding nucleotides, and these nucleotides are going to add RNA nucleotides rather than DNA nucleotides. Remember that I said DNA stands for what? Deoxyribonucleic acid. The deoxyribo part refers to the sugar portion, which in DNA is deoxyribose, but RNA is ribonucleic acid, and its sugar is ribose. It's just ribose. It's the same molecule, it's just that one is lacking in oxygen that the other one has. So the sugar is different, but the phosphate is the same, and the nitrogenous bases are almost the same, except in RNA, we have adenine, uracil binding to one another, and we have cytosine and guanine bonding to one another. So we replace thymine, in this case, with uracil. And so whenever you have an adenine in the DNA, in the original DNA, you're going to add a uracil RNA nucleotide to that. But otherwise, the complementary base pairing is the same. The other difference is that deoxyribonucleic acid is a two-stranded molecule, and RNA is a single-stranded molecule. So when you go to make RNA, you get this long linear molecule with nitrogenous bases exposed. Yeah.
Isn't the DNA not stable under I mean, it is stable, the RNA is not stable under what's it called, alkaline or something?
Yeah, where did you learn that?
I was learning this before or something like that.
Okay. Yeah, you're right, actually. RNA is generally not very stable. And this makes sense because what you're doing with RNA is you're creating it for a use. The reason that you're creating it is you want to make a particular protein, and so you create RNA, you use it to build that protein, and then you break that down. You don't want it hanging around because if you make too much of that protein, you might make more of that protein than you need. And so RNA molecules are notoriously unstable. As a matter of fact, if you study gene expression, the way you measure gene expression is actually by looking at the kinds and amounts of messenger RNA that are being created. And one of the difficulties of that is that in order to analyze that, you have to isolate the messenger RNA and convert it back into DNA before it degrades in the lab in your sample. So you do all of this under really cold conditions. You keep everything on ice because that slows down the degradation process so that you can keep that RNA around long enough that you can then get it converted into a more stable DNA form so that you can then go and analyze it. Otherwise, if you're not careful, it just degrades on you and you don't get any RNA out of your samples at all. And it looks like your organism isn't expressing any genes when in reality it is, and you just weren't fast enough to get to that RNA before it went away. Yeah, you're exactly right. So DNA is more stable than RNA because you have DNA, it makes RNA, you want to use that RNA to make proteins, but then as soon as you're done making those proteins, you want that RNA to go away and not continue to be used to make proteins. But this is a single strand molecule, and it has these exposed nitrogenous bases because those exposed nitrogenous bases are what specifies the order of amino acids that get put into this protein. So when we talk about protein synthesis, we use a cellular organelle called a ribosome. It's just big globular proteins that come in two pieces. The two pieces clamp around the RNA molecule going between them. And this ribosome is basically just a big enzyme complex. And its enzymatic function is that it picks up transfer RNA molecules, and transfer RNA molecules are also specified by DNA. The order of their nitrogenous bases is specific to particular ones. And so a given transfer RNA molecule will be specific to a particular amino acid, in this case, methionine. A different messenger or transfer RNA molecule will carry leucine. Another one will carry tryptophan. All of these amino acids, each amino acid has its own specific transfer RNA molecule. And so this strand of messenger RNA has sequence of nitrogenous bases. In this case, it's the adenine, uracil, guanine. So we have this messenger RNA strand, adenine, uracil, guanine. What's the next one? It's cytosine, uracil, guanine. And what happens is the large subunit of the ribosome and the small subunit of the ribosome clamp around this thing, but inside of here there are active sites. And these active sites are places where physically transfer RNA molecules come in here and reside. And the transfer RNA molecule that comes in here and resides here carries with it methionine, which is one of the amino acids. And the reason that it can fit in here is because it complementary base pairs with the codon, so in the messenger RNA molecule here, every 3 nucleotides is referred to as a codon, and this then is the anticodon, the opposite of the codon. So these transfer RNA molecules hook up to the messenger RNA, transfer RNA hooks up to messenger RNA through complementary base pairing, just like the two halves of DNA do. But this transfer RNA molecule is basically called transfer RNA because it's responsible for transferring amino acids from the cytoplasm to the ribosome where you're going to start to get proteins formed. The next one that comes in here, comes flying in from the cytoplasm, will have to have a different anticodon. What's the anticodon on this transfer RNA molecule going to have to be?
GUC.
Not GUC, GAC, there you go. Guanine, adenine, and cytosine. And that transfer RNA molecule has leucine on it. Once those things are in there together, what happens is the ribosome forms a bond between methionine and leucine, and then when it forms that bond with methionine and leucine, it kicks this transfer molecule out, transfer RNA molecule out. It goes back to the cytoplasm, picks up another methionine, and can be reused. And then this whole thing shifts down a site. So this whole thing moves, physically moves down the molecule so that it's now this transfer RNA molecule has moved from this site into this site, which means that there's now a place for this codon to specify a new amino acid. What's the anticodon going to be here? Guanine, guanine, guanine. Anybody know what that codon specifies for in terms of amino acid land? Amino acid language? Luckily, some scientists with way more time on their hands than is really allowable figured this out. If you know what the first base is, cytosine. First base?
For CCC.
I am looking for a CCC.
Proline.
And the
Yeah,
second.
I'm just trying to figure out. This is a wrong, what's wrong with this chart? This should be first base, second base, third base. Cytosine, cytosine, uracil. So switch these on this. I got this from someplace. So cytosine, cytosine, cytosine codes for proline. So there's a proline here. This proline attached to its transfer RNA molecule is going to come in here. What's going to happen? What's this ribosome going to do? It's going to form a bond between what and what?
Lysine and proline.
Leucine and proline. Once it does that, what's it going to do?
Transfer
It's going to kick that transfer RNA molecule out back into the cytoplasm. Whoop, that's CG, GAC. And then what is it going to do? It's going to move down another spot and do it again, and you get this amino acid sequence growing out of this ribosome. And it's attached to the ribosome through its connection to the messenger RNA, through this transfer RNA molecule to proline, to leucine, to methionine.
And it's just gonna keep doing that throughout the life cycle? Just over and
It's
over?
going to keep doing that until something tells it to stop. So here you have the most recent amino acid to be added, attached to its transfer RNA molecule, attached to the messenger RNA molecule. You have this long amino acid chain floating out here now in the cytoplasm, and you have the next amino acid ready to be bonded to that amino acid here in the ribosome. Yeah.
So what tells it to stop and why?
What tells it to stop is complicated, but ultimately what tells it to stop is these stop codons. So there are 3 stop codons: uracil adenine adenine, uracil adenine guanine, and uracil guanine adenine. When the ribosome gets to those transfer RNA molecules, the weird thing is that for these 3 transfer RNA molecules, uracil, uracil, adenine, and adenine, they don't carry an amino acid on them. And so they get to that point, this one shifts over to the new place, another one comes in here, and if it's this sequence, this amino acid, or this transfer RNA molecule rather, doesn't have any amino acid, so there's nothing to bond the previous amino acid to, and so that stops the process of protein synthesis. So these transfer RNA molecules are empty in a sense, and that they don't have an amino acid attached to them. So they don't code for an amino acid at all, they just code for telling RNA synthesis to stop. Yeah.
So instead of like the methionine floating around with the transfer RNA is like floating around trying to find a methionine, that UAA is just floating around to find its sequence.
So UAA will
Yeah,
complementary
UAA.
base pair here, but there's no amino acid, so there's nothing for leucine to bond to. So when it kicks this transfer RNA molecule out, releasing this, you just then release the protein. Because you've created this gap in the process, because there's no amino acid to bond to. So that's the simplest explanation. There are lots of upstream things that happen in the DNA and all that affect this, but ultimately it's fundamentally you're just missing amino acid on those transfer RNA molecules. This is how we go from a gene to the phenotype. What you look like, how you act, how fast you are in the 100-meter dash, et cetera, is we begin with genes. Those genes get transcribed into RNA, and in the language of RNA, the sequence of RNA nucleotides gets translated into a sequence of amino acids, and then those amino acids are basically the building blocks of proteins. And proteins are your enzymes, they're your muscles. Actin, myosin, all these things are proteins. The pigments in your eyes that give you eye color, those are all proteins. Those things make up a huge amount of what you are and how you behave, what your physiological capacities are, etc. And so that's ultimately specified by the nucleotide sequence in DNA, because that specifies the nucleotide sequence in RNA, and that sequence specifies the sequence of amino acids in your protein. We know which RNA sequences, which codons, code for which amino acids. So if we know the DNA of a gene, we can infer from that sequence of nitrogenous bases through complementary base pairing what the sequence of RNA will be. And because we know how RNA gets translated into amino acid sequences, we can use that sequence of RNA nitrogenous bases to tell us what the proteins are. So we can start with just a raw DNA sequence and predict what the protein sequence is that will come out of that, because we know these things about how DNA gets turned into RNA, RNA gets turned into protein. Questions? Okay, well, this is all hunky-dory fine until we start throwing in things like mutations. Mutations are just random changes to the nucleotide sequence of DNA. They're generally thought to be permanent changes, regarded to be permanent changes, and they can affect DNA that is in your nucleus. But also remember that chloroplasts and mitochondria, two cell organelles, also have their own specific DNA that is specific to those organelles, because mitochondria, which is where you make energy, mitochondria in you is where you take glucose, go through the process of cellular respiration to create ATP, which is what you get your energy from. So you eat plants that contain glucose, then your body in its mitochondria make ATP. Mitochondria are organelles. They're little things inside of cells. And then chloroplasts, we've already talked about. This is where photosynthesis occurs. we take carbon dioxide here and we make glucose. These two things, way back in evolutionary history, used to be bacteria. Sometime, way long time ago, some cells engulfed bacteria, and those bacteria didn't get basically digested by the cell. They became symbionts with the cell, and over time they became specialized to perform these two functions of cells: mitochondria in all cells and chloroplasts, which are limited to plants. And so when we look at the DNA that is found in mitochondria and the DNA that is found in chloroplasts, we find that that DNA is very different from the DNA that is found in the nucleus of the cell. Not only that, it looks like the kind of DNA that you find in bacteria. And so there's this theory called the endosymbiont theory, which is that two of our major organelles, mitochondria and chloroplasts, are actually just the remnants of this early symbiotic relationship that early cells had with these bacteria that they had engulfed. But it's a very different type of DNA. Our DNA occurs in long strands that have ends. Bacterial DNA is generally in a circle, so that there's no endpoint to it. So it's a double-stranded DNA molecule, but that double strand, it isn't arranged in chromosomes that are linear, it's arranged in chromosomes that are circular. Very different than the way the genome is structured in the organism that possesses the mitochondria that has the circular DNA. Remember, ours.
That mess with the health of the organ or the cells?
No. In that if you have a mitochondrial disorder, and some people are born with defects in their mitochondria such that they have a mitochondrial disorder, it messes with your metabolism because you don't have the ability to break down glucose and create ATP. So not having functioning mitochondria is a bad thing. Having functioning mitochondria is a good thing. So it doesn't have a negative effect on you having this. It has a positive effect on you having this. It's negative when you don't have this. So remember that our chromosomes are linear and they have an endpoint at each end. Bacterial genomes are circular generally. So when we talk about extra chromosomal DNA and RNA, we're talking about the DNA that are found in mitochondria and chloroplasts, a different kind of DNA that lives outside of the nucleus in these organelles. So lots of different kinds of mutations. You can have substitutions, deletions, duplications, what we call transposable elements. Transposable elements are just sections of DNA that tend to break off and go back and rejoin other chromosomes. The rates that this happen vary widely depending on the type of mutation, the gene of interest, the organism that you're in, et cetera, et cetera. So substitution mutations are often referred to as single nucleotide polymorphisms. They're referred to as SNPs in the business. So when you go home for fall break, you can tell your parents when they ask you, oh, well, what are you studying? Oh, I've had this ecology of food class. Oh, what are you learning there? Oh, we learned about SNPs. Parents will be like, what the heck is a SNP? Yeah. Single nucleotide polymorphism. It's just a bunch of fancy language. Polymorphism, just different forms. In this case, different forms of what? Different forms of DNA. Due to what? Single nucleotides, or really and truly nitrogenous bases. So it's just a fancy way of saying we took a gene sequence that was thymine, adenine, guanine, cytosine, adenine, cytosine, and we changed this guanine to a adenine. That's what a single nucleotide polymorphism is. It's polymorphic because it's a different form of DNA than it was before. And how is it different? It's different in this one nucleotide. Fancy pants language for something that's relatively simple. The thing about it is that some kinds of mutations happen faster, at faster rates than other kinds. And remember that cytosine and thymine are pyrimidines and adenine and guanine are purines. And because of the width of these, remember that a pyrimidine is what? In terms of, if I drew these two structures on an exam and said these are kind of the fundamental structures of a nitrogenous base, which one of these is a purine and which one of these is a pyrimidine?
Periods of top were correct.
No. Can you remember to tell these apart?
The number of sides they have.
Yep, so let's go back. Come on. Work with me, work with me. I changed this out.
Yeah, I thought it was different.
It was different, but it was blurry when I went to look at it, and so I changed it out to one that was clearer. Yeah. So we have two ring structures and single ring structures. The single ring structures, which are shorter, what did I tell you was a good way, or how I think about this? Yep, the short one goes with the long word. So these are pyrimidines, and these are purines. So whenever you change out nucleotides in a single nucleotide polymorphism, if you have a pyrimidine there and you put a purine in its place, you're now going to have a different width, and it's going to change the diameter of the molecule. And cells have a way of detecting those kinds of mutations, and they generally get rid of them because the molecule isn't the right size. And so we generally can trade out a pyrimidine for another pyrimidine or a purine for another purine much easier than trading out a purine for a pyrimidine or a pyrimidine for a purine, because when you do that change, it changes the diameter of the DNA molecule. So transitions occur faster. They occur so much faster that about 2/3 of all mutations, of all SNPs, single nucleotide polymorphisms, are transitions. Trading out one type of nitrogenous base for the same type of nitrogenous base. Pyrimidine for pyrimidine, purine for purine. The other third are called transversions, and this is when you have a purine being replaced by a pyrimidine or vice versa. Those change the diameter of the molecule, which is why they're less common, because they ultimately get filtered out by the cell's editing system. So the cell looks at its DNA, in a sense, it doesn't have eyes, but it has mechanisms by which it can look at the structure of the DNA molecule, and if that DNA molecule changes diameter, it basically will take out the nucleotide that is causing that change in diameter. It will actually stop the process of copying itself. It will take that nucleotide out, try another nucleotide in there to see if a different one fits better. And so cells have the ability to edit themselves as they're making copies of their DNA. Other kinds of mutations are more problematic. Go back to the codon table, and you'll see that a lot of times. In this case, serine is specified by anything that begins with a uracil and a cytosine. The third position here doesn't matter, can be anything, and it will still code for serine. This is what in the literature is referred to as the redundancy of the genetic code. When something is redundant, it means that you have two copies of the same thing and you don't necessarily need all of them. The redundancy of the genetic code allows for mutations to occur in this third position, and any mutation, any single nucleotide polymorphism that occurs in that third position, it doesn't mess up the thing that the code actually codes for, which is in this case serine. And so there's redundancy that makes single nucleotide polymorphisms not have very big of an effect. Additions and deletions, though, are different. Oftentimes these have big effects, because these are what we refer to as frame shift mutations. happens in a frame shift mutation is if this is the DNA sequence before the mutation, and we basically go through here and we get to a stop codon, so we have a methionine, a lysine, a phenylalanine, and a glycine. If we have an insertion such that it puts an extra adenine in right here, what it does is it shifts all of these things down and it converts this codon here that used to code for lysine, and it has switched it to a stop codon, which means that it stops the process of protein synthesis right after it got initiated. AUG is always the start codon. If we go back to our codon table, methionine, AUG is always the start codon. It starts off the process of protein synthesis by adding a methionine there. So at the beginning of every protein that you see that I'll ever give you, you will see that the first thing is always a methionine. In this case, by adding an extra adenine, by adding an extra nucleotide in there, it converted the codon that codes for lysine to the codon that codes for a stop codon. In the same way, if we remove an adenine, the methionine is still okay, the lysine is still okay, but then it changed this one from coding from phenylalanine to leucine. It changed that one from glycine to alanine. Because it shifted everything down a spot, every amino acid from that point on in that protein is going to be different. And oftentimes those different sequences of amino acids, they don't actually code for something that's actually functional. So it takes a functional protein and converts it to a non-functional protein. It doesn't have to convert this to a stop codon. It could have just converted this into a new amino acid that wasn't lysine, but then every amino acid from there on will also be affected because the frame of the codons, because the codon, remember, the codon is 3 nitrogenous bases at a time. And so if you have AUG, adenine, guanine, uracil, 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 when you get rid of one of these, the codon structure is now this instead of what it was before. That causes everything from where that mutation occurs, everything downstream of that gets altered. And that messes up genes. That messes up the product that the gene is specifying for it. It makes it non-functional. These things are referred to, these frame shift mutations are oftentimes referred to as nonsense mutations because it will still produce an amino acid, it's just the amino acid won't make any sense, it won't have any functional purpose. Questions?
What happens whenever there's a nitrogenous base that gets removed? Where does it go?
It just
Degraded somewhere?
Yeah, in the process of being copied, DNA makes copying mistakes, makes copying errors. In the same way that if you're copying somebody's notes, there might be errors in it as you go through and copy. You run things through a photocopier, sometimes the paper gets a little crinkled, and so there's a crease in it, and then when you get the crease out, there's a gap there. So copying errors are common. Mutations in general, a general walking around rate for mutations is 1 in every million DNA replication events, there's going to be a mistake. And so if you have 3 million nucleotides in your genome, every time you make a copy of your cell, you can expect at least 3 mutations in that cell because you had 3 million nucleotides to copy, and you get a mutation rate about every 1 in a million copyings. So if you have 3 million genes, that's three opportunities for mutation. So you went from a single zygote to an organism with trillions of cells. You probably have a number of mutations in you. Now, most of those mutations are silent mutations because they occur at the 3rd spot. of the codon. And oftentimes the third spot of the codon, changing that nucleotide, that nitrogenous base, doesn't have any effect on the protein that gets affected. Sometimes you go through a DNA duplication in the process of growing, and if you spend a lot of time out in the sunlight, ultraviolet radiation increases transcription errors, the rate of transcription errors. UV light is a mutagen, and what a mutagen is just something that increases the rate of mutation. And so this is why you get skin cancer, is because those cells are undergoing cell division, and if the rate of mutation is higher in those cells, the chances that you mutate your DNA into a type of DNA that causes cancer is higher, and that's why people have skin cancer, because we don't wear sunscreen because we're idiots. And so mutation rate can vary. In this case, it's an environmental thing that's causing the mutation rate to vary. And in many cases, those mutations don't have any effect, but in some cases, those mutations can turn into basal cell carcinoma or melanoma, and then you have to go get your cancerous lesions burned off. And so, did that address your question? Okay. Yeah. So, these are the kinds of mutations that have big effects. So basically what I've been taking you through so far is this thing that we call the central dogma in biology. All the central dogma says is that we use DNA to make messenger RNA, and then we use messenger RNA to specify the order of amino acids in a protein. This process of going from DNA to messenger RNA is transcription, where we use DNA as a template, and the process of going from the code in messenger RNA to the sequence of amino acids in a protein is referred to as translation. This is the central dogma, but this is how we go basically from genes that are found in DNA to the phenotype, which is determined to a large degree by proteins. So this is the big picture of all that we got. Because what we're going to do next is we're going to start doing genetics after we do some cell division stuff. Genetics, which is the process of going from genotype to phenotype, and then tracing how that gets inherited from the parental generation to their offspring to their offspring. The genotype gets passed down from the parents to the offspring, which then have different genotypes depending on the genotypes that their parents had. That then determines the distribution of phenotypes. And then those offspring can have offspring that also get their genotypes from their parents that translate into different phenotypes. So in order to understand how genetics is working, you need to have some knowledge of how we get out of a gene and into a phenotype. All right, questions? We have like 4 minutes left. We're going to talk about cell division next. We talk about mitosis first, and then we talk about meiosis next, because meiosis is easier to handle if you know about mitosis. And then meiosis sets us up for genetics, because it's meiosis where we create gametes, and gametes are where we get genes going from one generation to the next generation. So what we're going to do on Wednesday is we're going to blow through mitosis, meiosis, and then after that, probably on Friday, we will get to genetics per se, formally. So we're probably a full day to day and a half behind. So if you're looking at the schedule, go back and read tonight and go tomorrow night in preparation for Wednesday. Reread the sections that I assigned earlier on mitosis and meiosis, just so that you're up to speed on that for Monday. Have you guys all been exposed to mitosis and meiosis at some class in the past? Okay, I'm going to rely on you having read that in the book and reminding yourself about those things so that we can move through that pretty fast on Wednesday. Okay? And you know what? I have I'm going to have new food blogging prompts up on the website, on the Moodle site, probably within the next hour. So get back to Yes, yes, every 2 weeks. You have yours?
Well, I just don't have mine, so I don't like mine, so I don't care.
Yeah, put them in a little booklet, but give me what you have so far, because I need to.
I need to do the vlogging, I'm gonna put my name in there. Matter of fact, I can, I'm gonna just copy all this down and then, when I'm in notebook, I'm going to get one right now.
Yes, sir.
I've got mine right here for you too.
Oh.
I could have sent you about 20 pictures on Friday of it, but I just figured you wanted.
I would
right
prefer
here.
it this way, yeah. Okay,
Thank
I'll
you
look at
so
this and I'll
much.
give it back to you.
Okay, thank you.
Oh, yeah, sure, did it work?
Yeah, it did.
Cool.
Perfect.
Yeah, just hand them in to me that way and start this week with the stuff in a book.
Yes, sir. And, the blogging,